The disease we chose to investigate and report on is salmonellosis in humans. In this presentation, I will briefly discuss the history, causes of infection and symptoms, as well as the process by which Salmonella infects a host cell. In my conclusion, I will touch upon a recent development in Salmonella strain identification research from Penn State University. Salmonella enterica was discovered in the year 1885 by Theobald Smith, a scientist conducting medical research for the Veterinary Division of the United States Department of Agriculture. He was searching for the cause of hog cholera, a disease that plagued American livestock in the 19th century. Smith's discovery of the disease was obscured when Salmonella was named after Daniel Elmer Salmon. Salmon, the administrator of the department at the time of discovery, received credit for the discovery as his name appeared first on their published findings. There are over 2,000 serotypes, or strains of Salmonella, each one differentiated by its own unique genetic composition. Salmonella originate from the intestinal tracts of humans and other animals. Salmonella are typically transmitted via contamination with feces. Risk of contamination and subsequent infection by Salmonella can be greatly reduced or eliminated with the ease of an adequate hand washing. Salmonella are usually transmitted to humans via the consumption of contaminated foods. Contaminated foods are typically from animal products, such as poultry, pork, beef, eggs, or milk. Any food can become contaminated with Salmonella bacterium and will appear and smell normal. Food is often contaminated by the inadequately washed hands of the food preparer. Again, adequate hand washing with soap and warm water is a remarkably effective way to curtail or eliminate the chance for Salmonella contamination or infection. We'll now discuss the symptoms of salmonellosis. Most commonly, the symptoms of salmonellosis in humans include the following: diarrhea, fever, nausea, vomiting, headache, and stomach pain. Dehydration typically accompanies the vomiting and diarrhea. The most serious consequence of Salmonella infection is the risk of dehydration. In extreme cases of salmonellosis-related vomiting and diarrhea, intravenous replacement of fluids and electrolytes may be required by one's physician. As a potential treatment for less serious cases of salmonellosis, a physician might recommend antidiarrheal medications to bring temporary relief of stomach cramping to the afflicted. Antibiotics are another possible method for treatment, but are usually reserved for cases of suspected salmonella in the bloodstream. Symptoms such as stomach cramps, diarrhea, and nausea are often associated with other illnesses other than salmonellosis. Lab tests are required to positively identify Salmonella in the stool of a person that suspects they've been infected. Unless the salmonellosis is suspected to be systematic, many afflicted with the disorder recover from the related symptoms within or before a week's passing. If an examining physician believes that the Salmonella might be bloodborne, a blood draw and subsequent testing in a laboratory might be necessary for detection of the bacterium. Now we will discuss the process by which Salmonella infects a host cell. Imagine, if you will, a Salmonella approaching an intestinal epithelial cell's surface membrane. When it finally gets close enough, the Salmonella extends its type 3 injector system to inject effector proteins through the surface of the cell and into its cytoplasm. The epithelial cell membrane extends forward, engulfing the bacterium into the host cell's cytoplasm. Now that the Salmonella has infiltrated past the first line of defense, it is ensnared in a defensive vacuole and dragged in deeper by actin filaments. The typical defense mechanism would utilize autophagosomes and lysosomes to fuse with the vacuole and shower the invader with digestive enzymes. The type 3 injector system is used again by the Salmonella to modify the vacuole around it in order to prevent its fusion with and dissolution by defensive lysosomes. The vacuole containing the Salmonella then sprouts filaments linked with intracellular multiplication. Now, within the relative safety of its protective vacuole, the Salmonella begins to divide and divide, filling the vacuole to its absolute limit. Unfortunately, it is not presently known how the Salmonella bacteria move from the host cell to infect other cells. Now we will discuss the recent research out of Penn State University. Some recent research out of Penn State has found a new and improved approach to identify one of the 2,000 unique and separate Salmonella strains. Researchers at PSU's College of Agricultural Sciences have devised a method that focuses on virulence genes and two specific regions of Salmonella DNA on intermittent cycles. They describe the process involving the DNA going through clustered, regularly interspaced short palindromic repeats, or CRISPRs, C-R-I-S-P-R, CRISPRs for short. They subsequently designed a method they describe as multivirulence locus sequence typing, or MVLST. This process recognizes specific differences among the strains in the predetermined locations along cycling strands of Salmonella DNA. Some interesting facts about Salmonellosis. Did you know that in 1984, the largest recorded bioterror attack on United States soil was undertaken by the followers of Bhagwan Sri Rajneesh? They used an aerosolized form of Salmonella enterica cerevar tifimurium. His followers sprayed the bacteria on the salad bars of 10 restaurants in the Dalles, Oregon. Their effort was a move to neutralize the local electorate before an upcoming election of vital importance to their movement. Approximately 750 people were afflicted with salmonellosis as a result of the deliberate dissemination of the harmful bacterium. This concludes our presentation on salmonellosis and its causes, symptoms, and modes of infection. I hope you learned something new about a familiar disorder.